Next up, we have Dr. Myriam Missaoudine from the University of Montreal Research Institute, who will be talking to us about novel biomarkers in cancer immunotherapy from gut to lung microbiome. So we look forward to hearing this from you. Thank you.
So good afternoon, everyone. Thank you for the invitation, and I'm happy to share today our recent results, published and unpublished results on microbiome and cancer. So that is my disclosure. So as you all maybe all know, the gut microbiome represents a novel microbiome in IO and is now part of the hallmark of cancer. How it starts, actually, everything starts when our lab and others showed first in mice that the dysbiosis induced by antibiotics treatment induce an IO resistance in mice with tumor model known to be sensitive to IO. And in parallel, we showed exactly the same results in patients in different cohorts, specifically in lung cancer patients treated with IO, and we showed that the patients that did not receive any antibiotics treatment 1 month before and during their IO treatment respond better than patients treated with antibiotics. So these results actually were validated in several studies, and we are publishing a new meta-analysis in our lab, including more than 43,000 patients from more than 100 studies, showing exactly the same thing and validating this deleterious impact of antibiotics on IO and chemo response. So in addition to the importance to maintain the integrity of microbiome, the gut microbiome, the composition of this gut microbiome is important. And actually we showed that, for example, the presence of Akkermansia can correlate with the IO response in lung cancer patients treated with anti-PD-1. And actually the presence of this Akkermansia and increase of abundance of this bacteria is correlated with a better IO response in lung cancer. So actually following all these results, we can observe since few years now a rapid evolution of the gut microbiome in oncology. And this evolution is correlated with the numerous clinical studies that they are currently ongoing in all around the world. And we have some of them at the CHUM, and actually with the collaboration of with all the oncologists at the CHUM, and also in Montreal, at the Jewish hospital, but also in other regions in Canada, such as London, Ontario, Sunnybrook, de Quebec. And here, I listed actually the clinical trials portfolio that we have at the CHUM Microbiome Center. We have several clinical trials with the fecal microbiota transplantation when we transfer feces from donors to cancer patients, specifically lung cancer and melanoma patients. We have also clinical trial in collaboration with biotech, and also we are testing prebiotics, and we are also evaluating diet and its impact on microbiome composition and the IO response. So I will start, and then we summarized what ongoing with the FMT clinical trial. And actually, since 3 years now, there is a first in human studies demonstrating a very strong potential of FMT to reduce the primary and secondary resistance to immunotherapy in melanoma patients. The first 2 clinical trials, they included patients treated specifically in second line with FMT plus single agent anti-PD-1. And the third studies that we had actually at the CHUM, MIMIC clinical trial, we included patients treated as first-line FMT plus single-agent anti-PD-1. It's a first Canadian FMT clinical trial And in these studies, the results we were able to increase the response by 20% compared to randomized clinical trial, phase 3 clinical trial, when the response of the patients treated with only anti-PD-1 is around 45%. So after the results of MIMIC clinical trial, we opened a second clinical trial, a phase 2 FMT Illuminate, when we included 2 different cohorts. The first one with advanced non-small cell lung cancer patients treated with anti-PD-1, and the second cohort with advanced melanoma patients treated with anti-CTLA-4 and anti-PD-1. We finished the inclusion of the patients in this clinical trial. I can show you here the preliminary results for the best overall response. And for the lung patient, for the advanced non-small cell lung cancer patients, we observed the ORR of 78.5% with an increase of around 40%, 35% of the response in these patients compared to phase 3 randomized phase 3 clinical trial when the response is around between 40 and 45%. And for the advanced melanoma patients, we observed an increase around 20% of the response in this cohort of the patients. So following these preliminary results with this phase 2, we are actually opening a randomized phase 2 clinical trial when we will include melanoma patients, and we will include 128 patients in collaboration with CCTG and founded by CHR. So here I talked about advanced non-small cell lung cancer patients, but now we know that the neoadjuvant chemo-IO is the new gold standard in limited non-small cell lung cancer patients. So we want to study the microbiome, the gut microbiome, but also the local microbiome, the lung microbiome, and the intratumoral microbiome in patients with limited non-small cell lung cancer patients treated with neoadjuvant chemo-IO. So for that, and actually just to go back to these questions of intratumoral bacteria and the local microbiome, it's a very controversial subject because everyone is not, we don't have all the same opinion about this field and this subject, but there is very strong results with several studies showing that the tumor is not sterile and may influence response to cancer therapy. And we have an in-press paper showing that the presence of Escherichia coli in the tumor, within the tumor, can influence the response. All the tumors positive to Escherichia coli respond better than the group without Escherichia coli in the tumor. So we want to study this local and intratumoral microbiome. So we have 3 different axes to study that. The first one is to analyze the microbiome, the bacteria and the microbiome in the bronchoalveolar lavage. We are collaborating with Dr. Lieberman at the CHUM to collect this BAL, and we have already collected 42 patients. We want also to analyze using NGS sequencing tumors, non-small cell lung, limited non-small cell lung cancer tumor treated with neoadjuvant treatment. And also we will collect for all these patients the stools of the patients to analyze the gut microbiome using the metagenomic sequencing. So here I can show you the very preliminary results for the BAL, for the bronchoalveolar lavage of limited non-small cell lung cancer patients. We used 2 different techniques, the 16S sequencing and the culturomics. And actually both of them gave us very interesting results. We can see that the BAL from cancer patients they have actually a higher abundance of some bacteria, specifically Streptococcus bacteria, and also a complete different composition compared to the BAL from patients with the cancer-free. So to conclude my talk and to summarize the different points, so the gut microbiome represents a novel biomarker of IO and is now part of the hallmark of cancer. We need a pan-Canadian microbiome profiling study to identify a signature of response to IO and IO plus chemo, and to develop better strategies and better therapeutic strategies to modify them favorably the microbiome. And actually, we have already several strategies that are currently being evaluated in clinical trial in Canada and at the CHUM and also in other hospitals in Canada to improve microbiome composition and IO outcomes. And now we have projects to study neoadjuvant treated limited non-small cell lung cancer patients. And we are trying to determine the link between the local and the gut microbiome and the treatment response of these patients. And this part of the project is funded by Terry Fox Marathon of Hope. So, and finally, I would like to thank all our collaborators, our funders, and also my team. Thank you.
Any questions for Dr. Messaoudine? Yeah. Maybe while the microphone gets there, maybe I'll ask you, to what extent do you think it matters that we're getting an actual bronchoalveolar lavage to sample versus just sputum? And does that make a difference in your view?
We don't know.
We don't know yet. Sorry.
Oh, sorry, we don't know.
Yeah.
Not yet.
Not yet.
We are trying actually to find the best way to analyze actually this local microbiome. And the first one, it's the easiest way to do it. It's to have access to the BAL.
Sure.
Yeah, but yeah. I don't have a very clear response for that.
Yeah.
Well, it was a great talk and very interesting. Can you show me your second slide? The second slide. It's just that I think your point was there's this relationship between antibiotics and the role of antibiotics and the decreased immune response. And I think clinically, we don't actually see that. We have all these smokers that get COPD exacerbations. The first thing they do, the respirologist puts them on steroids and antibiotics. We don't really see a decline in their response to the immunotherapy that we're giving.
Sorry, I didn't hear you, but sorry, I'm sorry.
So the question is, Barb doesn't see in her clinic what she's showing in her graph. So who's right?
Actually, here for the patients, you mean in the meta-analysis? The patients included in this meta-analysis?
Well, I may help answer. So, your opinion and all the studies that show otherwise. So every study that we've looked at so far, looking at antibiotics before IO, gives bad results. So it might be because of the patient, it might be because of the antibiotic, but there's certainly a prognostic factor in needing antibiotics.
Okay. Thank you very much. That was fantastic. We're going to maybe move on to the next talk just for the sake of time.